Good morning, good afternoon, and welcome to MPN Research Foundation's live webinar with a presentation of our most recent poster abstract titled Characteristics and Features of Non-Caucasian Myeloproliferative Neoplasm Patients in the MyMPN Registry. Just a reminder that this presentation is being recorded for future viewing via the Foundation's YouTube channel. For anyone unfamiliar with MPNRF, we are celebrating our 20th anniversary this year. In the last 20 years, we have funded over $15 million in research, including projects which led to the discovery of the CALR mutation and major initiative focused on interferon and its effectiveness as a therapy for MPN patients. 3 years ago, MPNRF launched MyMPN, an online registry for patients to contribute their individual diagnoses, symptoms, and health events. I am the project manager for that. It's called MyMPN. The data is de-identified and made available for research, clinical, and pharmaceutical community to improve treatment of patients and move therapies forward more rapidly. We'd like to acknowledge at this time the support of our pharmaceutical industry partners, Celgene, now Bristol Myers Squibb, Prometeor, which is now part of Genentech Roche, and also Pharmacentia. We are very grateful to them for their support, which enables us to continue this important work. The annual Congress of the European Hematology Association typically draws about 25,000 clinicians, researchers, pharmaceutical reps, and some advocacy organizations. This year, the event was held virtually with video presentations of the latest research and drug trial results made online over the last week. Given that everything is virtual at EHA this year, we decided to open up the event to the entire MPN community, including patients, researchers, and clinicians. We're glad you decided to join us. In a moment, I will introduce our speaker and panelists. First, I would like to remind you that all participants, other than speakers, are muted for the duration of the event. If you have questions, please submit them via the Q&A below. On your screen below in the Zoom function. Kindly keep your questions brief, do not share personal medical details, and stay within the theme of MyMPN registry and the use of its data. Our panelists today include Dr. Serge Verstovsek, who is a medical oncologist and professor at the Department of Leukemia at the University of Texas MD Anderson Cancer Center in Houston. He has been a practicing physician focused on MPNs for over 7 years. Many of you know Dr. Ruben Meza. He is the director of UT Health San Antonio MD Anderson Cancer Center. He has been involved in MPN research for more than 20 years and we're very grateful for him for his leadership as the chair of the steering committee for MyMPN. I'm going to turn it over to Allison Moliterno. Allison is a researcher and, as a researcher, hematologist, and associate professor of medicine at Johns Hopkins University Medical. In Baltimore, Maryland. Dr. Moliterno maintains an active hospital-based clinical practice focused on the optimal care of individuals with myeloproliferative neoplasms and other hematologic conditions. And she also serves as at the hospital as an attending physician for both hematology inpatients and consultation for a broad range of hematologic conditions. Her basic and clinical research areas are focused on elucidating the molecular underpinnings of myeloproliferative neoplasms and developing effective treatment strategies. Dr. Malone-Sorno, thank you very much for presenting these slides. I'm going to turn it over to you now.
Thank you, Lindsay, and it really is an honor and a privilege to be with everybody here all together to share the accomplishments of the MPN Research Foundation and particularly the MyMPN Registry. I'm presenting this on behalf of really the tireless efforts of the MPN Research Foundation staff and patients and researchers who have all really made this registry. And particularly, I would like to acknowledge Dr. Robin Sherber, who put these data and the presentation together for the EHA poster session and who was not able to present it for us today. So the MyMPN is a really very powerful tool for everybody. This was launched in September of 2017 and currently has over 1,200 participants in the US. All states are represented, but we do have the highest participation in residents of California, Texas, and Florida. The purpose of MyMPN is really to do many things that hopefully, as this diagram shows, will feed into each other, to establish a community, to learn about disease prevalence and progression, to help drive research goals that are in the interests of our patients, to study best practices in care and treatment, and also to be an avenue to recruit patients for trials and to broaden the awareness, the situational awareness of MPN therapy. So these are all goals that I think have already started to become evident in just the few years that this has been available with such great adoption of this registry. And this is translated to now starting to look at the data, shape it, and present it. And this was done at the European Hematology Association in 2018 and 19, and also the American Association of Cancer Research last year, and then the MPN Specialty Congresses in New York in 2018 and 2019. So the importance of real-world evidence is manyfold. Partly it's due to real-time analysis. So what we understand about disease is when we look backwards into textbooks or into published data, that's really looking backwards in time where things may be not relevant today. So having this real-world and real-time evidence allows us to make hypotheses about treatments and incorporates new data, genetic profiling, the patient symptom experience, previous responses to therapies, and other measures and combinations of data points. This also, by generating these data and asking questions, the pharmaceutical industry can also help identify potential participants in trials, and clinicians and patients can use findings from the registry as guidance in treating patients and understanding disease. What I wanted to show here was the success of the publications that we've had so far. Prior work with the registry, as I mentioned, has been shown in international meetings, and shown here is a poster presentation from October 2019. And what we want to show here is how we didn't know how strong the data coming into the registry from patients would be, this study shows that it's actually very reliable and very strong. This study examined both the mutation prevalence and correlation of mutation with symptom burden. Next slide. And what you can see here on a blow-up of the poster is that 583 patients entered data, and two findings emerged. First was that really the same distribution of mutations were evident in the registry as were been published in large academic cohorts. And also there was a very significant difference in age at presentation for CALR individual positive patients compared to JAK2. Next slide. When participants also correlated their MPN 10 surveys and symptom burden, and this was then delineated by mutation burden, mutation type, and you can see that there was significantly increased evidence of a higher symptom burden in JAK2 positive compared to CALR mutation positive individuals. individuals or MPL positive individuals. All right, next slide. So this was really a first analysis to identify that genetic mutations may be a contributor to specific symptom phenotypes. So again, it really is very important what your underlying lesion is. And it fits with the age and more indolent clinical course of CALR mutated MPNs that's been promoted or described in large academic reports. So we conclude that the data in MyMPN mirrors closely with previously published and validated MPN populations. And so this is very exciting and important because within really just a year or two, we got several hundred patients in, and now we realize that these data that we're generating from this have a lot of validation and integrity. So on to today's poster. The title of this was Characteristics and Features of MPN Patients in the MyMPN Registry. And again, the goals were to investigate possible health disparities in the registry representation and to consider an advocacy for a solution-oriented paradigm for differences and brainstorm solutions also to increase participation and representation in MyMPN. As of September 2019, 981 individuals 968 individuals had entered data to the registry, and this was the cutoff to analyze these data. 54 individuals were underrepresented minorities who participated in this, representing 5.6% of the MyMPN cohort. So conversely, Caucasians represent 94% of the MyMPN registry, and that's delineated here in the first column of the MyMPN registry, the breakdown of the racial, self-reported racial and ethnic. Interesting though, the Caucasians represent less than 90% of MPN overall. So if you look at published data sets, and we published one recently in Blood Advances, really only about 84% of MPN overall in our registries are Caucasian. And so that this, the MyMPN registry actually has an overrepresentation of Caucasians, or therefore an underrepresentation of unrepresented minorities. And then on these two columns are the US population in California and the US population in Florida. And you can see, of course, in the general US population, not an MPN population. Again, Caucasians roughly are 40 to 50% of the population, and where underrepresented minorities comprise a much larger proportion of the population. So, and these differences, I'm sorry, the previous slide, were most prevalent in the Hispanic and Latino group. So again, roughly 20 to 23% in the US population. And this was very underrepresented in the MPN registry. So the lack of representation of minorities in large US MPN academic reported cohorts has really required explanation. And this may be due to genetics or predisposition to MPN that differ across racial or ethnic groups, but also may be related to factors due to patient access to clinical trials or research engagement and many other factors. We have an even lower representation in the MyMPN registry, and so we have to have some thought there and investigation on how we can engage more underrepresented minorities to join the registry. Okay, next slide. When we drill down to the 54 individuals who are underrepresented minorities, we can look at the demographic features to understand if there's differences in disease presentation. We find that Hispanic and Latino do make up the largest percentage of underrepresented minorities of this group, and that's true in the US population also. With regard to age and gender, the underrepresented minorities mirror MyMPN at large cohort and other US MPN cohorts, with about 60% of the cohort being female and the age in the mid-50s. So that checks with the group at large. All right, next slide. MyMPN also captures a very granular information that is important to healthcare literacy. and which is critical to outcomes. Education is a very important factor in healthcare literacy and outcomes, and this information is captured in the registry. As you can see here, the 54 MyMPN underrepresented minorities had very similar education levels, with the majority of individuals having college and advanced degrees. And this was very similar to the educational level of the entire registry. MyMPN also captures patient-reported disease features, including disease type and disease transitions. And below you can see the distribution of the three most common disease types: essential thrombocytosis, polycythemia vera, and primary myelofibrosis. And the cohort at large in blue bars here, and the underrepresented minorities in the orange bars here. You can see there that in the underrepresented minorities, there's a higher prevalence of myelofibrosis and a lower prevalence of PV compared to the cohort at large. Next slide. MyMPN also captures patient-reported disease features, including genetic drivers or mutations that cause MPN. Shown here are the most common mutations: JAK2V617F, CALR mutation, JAK2 exon 12, MPL, and also patients who were unaware or it was unknown what their driver was. Like in prior abstracts and reports, oh sorry, could stand that, yeah. Self-reported mutation data mirrors large academic reports, with JAK2 comprising 70% of the mutation burden for the group, CALR about 15%, JAK2 exon 12 about 5%, and MPL the least common mutation. So again, the underrepresented minorities have similar distributions here in orange compared to the cohort at large, with perhaps a slight excess of JAK2V617F compared to the cohort. MyMPN captures current therapies, and that is listed here. Most common treatments, hydroxyurea and ruxolitinib, are most common in both groups, and you can see here, hydroxyurea is the most common therapy for both groups at around 30 to 35%. Ruxolitinib also about 12 to 15% for the groups. There is a trend in this of patients reporting no therapy or aspirin alone, and 23 of 52 individuals reporting no therapy who are underrepresented minorities, where this only occurred in 32%. There's a mistake on that side of 251, 187 individuals, or 32%. So that's an interesting finding that there does seem to be a slight difference, excess of no reported therapy in the underrepresented minorities. And then finally, MyMPN also captures settings of care, which is also an important determinant or description of healthcare utilization and outcomes. Here we have care divided, whether a patient receives their care at an academic medical center or university, a large or regional tertiary medical center, or the local community hematologist oncologist office. And significantly here was that individuals who were underrepresented minorities had a higher prevalence of receiving their care at academic medical centers or large tertiary care centers, as opposed to local or community hematologist office. You can see the difference here, 18% versus 40%. And this was highly significant. All right, so in summary, this is the first analysis of racial minorities in a patient-reported registry of MPN. The MyMPN registry has shown the integrity of patient-reported data that is in line with what we understand about sex differences and mutation burden, for example. However, Black, Asian, Hispanic, and Native American Alaskan comprise a much lower percentage of the registry than expected from prior MPN reports, and also from the US population. So we really need to understand the basis of this. Underrepresented MyMPN registry participants may have higher burden of disease, higher JAK2 mutation rates, and yet seem to have lower rates of therapy. indicating perhaps barriers to MyMPN participation and patient treatment needs study. We found significant differences also between where individuals receive their care, with underrepresented registry participants receiving care more often in academic or tertiary care facilities. And finally, we're very excited to further mine these data collection ongoing regarding outcomes, and particularly thrombotic risk, which may be previously noted to be high in underrepresented MPN patients. So I hope that we've shown you that the MyMPN registry is really an ongoing, extremely valuable and critical resource for patients, for patient scientists, for physicians and physician scientists, caregivers, industry, and anyone who's really interested in MPN disease, factors important for disease evolution, outcome, and treatments. I really would like to thank you for your attention to this presentation, and I'm happy now to turn this over to Lindsay, right? Is this where we turn it back over, Lindsay? Or not? Oh, there you are.
Hi, can you hear me now? So if you are a patient, please do consider joining us on MyMPN. If you are already a participant in MyMPN, thank you so much for your participation, and we encourage you to come back often. If you, have recently visited the doctor, if you have some sort of a medical health event that you'd like to report. I hope that some of this presentation has illustrated the importance of that data and, how this concept of a longitudinal data collection is so valuable. We'll talk a little bit more about that for sure during the question and answer period. And for any researchers or representatives of the pharmaceutical industry who happen to be on the call, we welcome you. Thank you so much for joining us. And we want to let you know that de-identified data is potentially available for your studies. If you would like to learn more, there's a link here, but you can always contact us at the MPN Research Foundation for more information. And we'd love to talk to you about how we can support your research. So now I'm going to hand it over to the panel of members of our steering committee. When I was doing the introductions earlier, I did not realize that we did have Amy Luduek also on the line. And so if we can make sure that Doctors Moliterno, Meza, Verstovsek, and Duek are all unmuted, and we can begin with some questions and answers. I'd like to reiterate that questions should focus specifically on MyMPN, the registry, any of the data that's been presented in this presentation, this event today, and how that data and the registry generally can support the development of pharmaceutical therapies and the better understandings of MPNs and the progression going forward. Please do not share any personal medical history. We won't be able to address any questions about anyone's specific situation. And one last time I'll mention, at the bottom of the screen in Zoom is a function that says Q&A, and that's where you can submit your questions. So now that I think we're ready to kick it off with the Q&A. So the first one is from Ned Sanger, who was actually a participant on a recent event that we had in Washington, D.C. for the FDA. It's good to see you again, Ned. The question is, given only 1,200 patients have registered in MyMPN, whereas there are probably a total of approximately 330,000 in the US, how significant are these results? It does not seem to be highly powered. Dr. Meza, would you like to tackle that one?
Sure. So Without question, the broader the representative sample, the more that we're going to be able to draw both observations and conclusions. MPN patients are very heterogeneous, both in terms of age, gender, disease features, but obviously individual features such as race, ethnicity, cultural values, and many other pieces. Clearly, the richer the sample, the more that we'll be able to understand. And indeed, these differences are very important for many reasons. First, differences in terms of biology, differences in terms of symptoms, risk of blood clots, mutational profile, potential risks of disease progression, the ability to react to medications, toxicity from medications, and longer term outcomes. So many pieces. So without question, that's a great lead-in question that just to further encourage everyone who is not participating to consider being part of this registry. It's a very unique resource, and that's one of the reasons I've been excited to be working with the MPN Research Foundation and all of my colleagues in generating this, in that we hope it really has a very broad base. Many of the publications and data that we have used in our field in MPNs have largely been derived from single centers. And although those have great value, they certainly sometimes lack the breadth of characteristics that we would like to see. Indeed, as we see the data even from today, the fact that the majority of the minority or non-Caucasian patients that have participated in the registry are from academic centers is probably not by accident. It probably means that those individuals, again, are more likely to feel that they're connected to the online MPN community and have heard about Be My MPN, while others in community practices may feel quite a bit more isolated in terms of their experience. So we still have work to do to continue to spread the word, to get the broadest and most representative sample of MPN patients to be able to learn and share those experiences.
Great, thank you, Dr. Meza. Does anyone else have anything to add to that? And if not, I'll go ahead to the next question. We did have a question come up separately on the chat, and I think it works nicely with another question that we had from prior to the event. So given the focus is on minorities in the registry in this particular study, how are we ensuring that all medical research is including enough data and participation from non-Caucasians living with MPNs? And the comment in the chat was that there will be potentially bias. This individual mentioned specifically Asians do not tend to share medical information. So I wonder if maybe Amy Lou, would you like to, Dr. Dueck, would you like to tackle that one?
Sure, sorry, I'm like reading the questions. I might have missed the very end of what you just said.
Yeah, this one actually came in prior to the event. I'll read it again for you. Given the focus in this study was on minorities in the registry, how are we ensuring that all medical research is including enough data and participation from non-Caucasians living with an MPN? And then, like I said, this particular focus of this individual in the chat was on Asians who tend not to share their medical experiences.
Yep. So it's a fantastic question, and certainly an issue with much of the clinical research and clinical trial participation that we do. So an underrepresentation of folks makes it very difficult to answer specific questions. But from a statistical perspective, as long as we have some folks participating, we can rebalance or recalibrate estimates to reweight information so that it can properly represent, say, an entire patient population, even if we still have slight underrepresentation. Those are some of the things that we're planning to do with this data. You know, but we do need, you know, we still need some folks to participate, even if it's a small number. And you know, so it's definitely very important to try to get as much representation and as many diverse folks participating, so that, you know, we have that data and that information to be able to produce estimates that can represent the entire population.
Wonderful. Again, anyone else on the panel, any thoughts on that topic? Okay, here's an interesting follow-up. Are there thoughts on follow-up research from this data? What kinds of questions does this provoke? Dr. Prostovsik, perhaps?
I'd like to build on the prior comments, particularly
Sure thing.
Dr. Meza's comments, where the notion that the patients that are minority participate, if that is suggestion from the results, if they are taken care of in the large academic centers or tertiary hospitals, gives us a thought that the widespread understanding of the disease process, of education, what the disease is all about, what the therapies are, what the diagnosis means, and what the options are there for patient education, patient participation, perhaps is not widely spread in the minority communities. In particular, here in Texas, where we are, Ruben and I, we have been discussing efforts to engage more hands-on with the local doctors in community settings, not to only advise on what the patient should be treated or how, and provide guidance to the doctors, but to engage with the patient's communities and enhance the participation of minorities, Hispanics in Texas, obviously, to bring that large population, as it is in California, for example, as well, to be represented properly, and so for us to understand possible differences at all these levels, from diagnosis to prognosis to therapy and to education, as we are talking about this right now, to a larger group of people that are underserved or underrepresented in the registries. As you said in the summary slide, this is one of the first efforts to see in a large patient registries, to see what is happening with minorities, and we are only at the beginning here. And so it's very interesting effort here, and gives us a lot of things to think about, but to act as well. And we may start first here in Texas on a ground level and build it up with your help.
I'd like to make a few comments also. Thank you, Serge. Those were very helpful. But I think all of us now living through COVID have really seen how much digital access and access to the internet has been critical in getting care, delivering care, and that's a huge social determinant of health now, right? Getting this access. I don't have time to educate my patients in clinic, but I do have time to point them to the MPN Research Foundation for further education and so I think this also really highlights how important it is to provide this access to individuals who may not have used this in their healthcare before. So I think this really shows us how important moving forward for all of us to really engage the community and in this fashion.
Wonderful, thank you. I have a question in the Q&A section regarding secure. How do you secure the privacy of my data? So I can take that first, if it's okay with the panel. I would like to just point out that the registry is completely voluntary. When you initially sign into the registry, you have the opportunity to designate how you would like your data to be used. In fact, if you just want to use the registry for your own data repository purposes, where you keep track of your symptoms and your health events, etc., you can do that. but the registry also, in the privacy settings, which is where you're directed when you first sign on for the first time, invites you to make your data available on a de-identified basis to many different segments of the research and drug development sort of community broadly. So you can choose to share your data with researchers only. You can choose to share your data with researchers and pharmaceutical companies, et cetera. But I have to emphasize that all of this is on a de-identified basis, that your identity would never travel with any of the information that you provide us. And I would welcome any members of the panel to add to that.
But perhaps I'll comment that one, I could certainly attest that the MPN Research Foundation has really made, privacy and the protection of the data, the highest priority from the beginning, kind of the conception of the study, and then that's further validated by the institutional review board that, acts as kind of a steward and oversight in terms of the study. So we're very mindful of those parts, and perhaps Dr. Dewitt can discuss that as well, but how, the lengths that are undertaken to be certain that we're able to look at data really in a de-identified way, but not in a way that really would be a risk to any individual patient.
Yeah, and I can just confirm that we certainly look at consent when we're using the data, and that would be true in all the research that we do. But particularly for this registry, we would absolutely be considering consent in any data that we use. And in presentations, we generally show summary data for groups of patients, and so we typically don't ever show just individual lines of data in any presentation. So that also adds further anonymity to the information that you're providing. It would be it would be essentially impossible to dissect from any presentations that we do, who was present and who wasn't present in any given analysis.
Very good, thank you. I thought perhaps we might turn to some questions that have been submitted in the past prior to the event today, actually via email, that focus on the development of therapies for MPNs. And so we had a couple of questions come in about specific diseases, so myelofibrosis and polycythemia vera specifically were mentioned, but I think generally speaking, the questions focus on how can we measure the progress of therapies for MPNs, and why do some patients progress more quickly than others? And what is the status of new therapeutics? So it's kind of a large sort of category of questions, and perhaps what we could do is just kind of go down the line, and each of you could comment on how you see MyMPN's role in this process of better identifying progression of MPNs, as well as the development of therapies and better treatments. Shall we start with Dr. Moliterno?
That's a lot of big questions there. I think the registry will be some of these other questions related to lifestyle, health conditions and lifestyle factors. And those are very important in understanding why some patients progress more quickly than others. And so I do think the registry will be able to inform that, because we are capturing disease transitions and progressions. I think it is very difficult to look backwards as academic researchers, as we have to do, to look backwards to say, well, these percentage of patients progressed and what were those factors. So I do think that the registry can inform that research prospectively.
Okay, any other comments? Dr. Meza, would you like to tackle that one?
Sure. Well, so one, I could say that, I'm incredibly hopeful. Having been in this field a long time, along with all of my colleagues, we've really seen a transition where there were no MPN specific therapies and we had relatively limited knowledge regarding their biology. And we've learned a tremendous amount, both in terms of how genetics and genetic changes lead to both the initiation of MPNs, and we certainly a lot of focus on understanding, why do they evolve? And then secondly, many different pharmaceutical companies that really have prioritized developing NPN specific therapies. So all of that really has occurred over the last, let's say, 15 years, where before that it really was, much more vague. The role of the registry, as well as I think the discussions that the MPNRF led, with the FDA in the fall, is really showing the heterogeneity of MPN patients, and many of the areas of unmet need. It's a period of tremendous advances. So concurrent with this presentation, this occurs, as was mentioned at the beginning, was part of the European Hematology Association meeting. So just to give you a bit of a flavor of the advances being discussed there, they ranged from a study by our colleagues in Italy and Tiziano Barbui, looking at ropegulated interferon versus just phlebotomy and aspirin for low risk PV. Potentially, if that continues to be positive, potentially, altering some of that paradigm for PV, better control the disease, better control symptoms, maybe decrease risk of thrombosis, maybe decrease the risk of disease progression. And many different therapeutic advances being discussed. Even indeed this morning, there was live question and answer on multiple new types of therapy, as well as concurrent posters, from drugs being used in isolation, other JAK inhibitors such as momelotinib, pacritinib, updated data with fedratinib, clearly very mature data with ruxolitinib, many combinations, ruxolitinib with PI3 kinase inhibitors, with the BET inhibitor from Constellation Pharmaceuticals, the new therapy from Imago Biopharma, just to name a few. So tremendous progress. But when we say MPNs, we are really thinking of a very broad group of individuals. So what might be a goal therapy for a young patient with ET might be very different than a 78-year-old that has advanced myelofibrosis. So the diversity and just the depth of research going on is something to be incredibly hopeful.
If I can add a little bit on the utility of the registry in terms of understanding the therapy that is currently being done or used. And Ruben has mentioned interferon. So in this database, we see 5 to 6% of the patients on interferon. I have seen some other larger databases where the percentage was even lower, 1 or 2%. So understanding who are the patients in our database here that are receiving interferon. Is it more in PV or ET or any myelofibrosis? What are their ages? Is it the disparity there present between the different patients on different characteristics? Where do they get that interferon? Who are the doctors that are prescribing it? Is it in a large tertiary centers? Is it the local community? Is it more in one state than the other? Is it related to the insurance issues, for example? It's very hard to prescribe it here in Texas. It's not so hard in New York State. How long does it last? How long do they stay on interferon? So we can learn from registry a lot about current state of affairs and build on that with the new medications like Ruben mentioned, ropeg interferon, that possibly is coming from Europe to the United States in very near future. So this is the alternative aspect of the benefit of having registry and following patients throughout their life with the disease.
Wonderful. Thank you. Yes, I actually really appreciate that you brought that up, Dr. Meza, about the other presentations that were going on at the same time during EHA. In fact, one of the ones I found most interesting and potentially something that we could relate to in the NPN space was a presentation specifically about precision medicine. Would you say that precision medicine could benefit in the long run? Can we potentially use something that comes out of the registry as we think about how to treat certain cohorts of patients? Perhaps one of you could outline how we can do a study that would maybe focus on some genetic mutational aspects or some other aspects that that could help move a therapy forward, or help us understand why, to Dr. Verstovsk's point, why some therapies might work better for certain patients than others.
I guess I can start. You know, when we do clinical trials, they're typically powered, or we include enough patients to answer. Usually it's the overall question of whether a drug is effective or not, versus either kind of standard of care or some other competing therapy. But generally speaking, within just that clinical trial, there's not enough statistical power to dive deeper into questions like, are there specific subgroups that, say, receive more benefit than some other subgroup? And so, you know, typically there's many questions that go unanswered in our clinical trials. And it's really kind of post-approval or after these drugs start getting used out in the community that we can start looking at data to understand, are there subsets of patients that do better? So maybe they have fewer symptoms than other patients. They might be receiving more benefit from a particular drug over another. And a lot of questions that allow for more kind of personalized, you know, administration of drugs. But, you know, you generally need a lot of data to answer those questions. And that's where I think, you know, registries and observational data can be very helpful to, you know, to both develop hypotheses, potentially, you know, test hypotheses within the observational data, and then move them into other clinical trials to formally test it in that setting as well.
Wonderful. Yes, I think it really speaks to the heterogeneity of MPN patients that's been mentioned a couple times previously. So that's good to know. Let's see, we've of course had a few people mention COVID, and I know that it was mentioned earlier, but I thought perhaps we might just want to touch on that topic briefly. I wondered if anybody had any thoughts on patients in the registry that have experienced something having to do with COVID. Is there a route for them to go? At the present time, we don't have a survey in MyMPN for patients with COVID, but I think that there are some things going on out there, and maybe if we wanted to just take a moment to, if you're aware of some other options for patients, then we can, of course, help spread the word about that through the MPN Research Foundation as well. But Dr. Meza, did you have something that you would like to share about COVID and any guidance for patients that are perhaps recovering?
Sure. So first, of course, there's not a patient out there that doesn't have a question as to how COVID relates to them. Completely natural, as it's a question that relates to each of us. We are trying to gather data. Dr. Duick and I and many colleagues are working together on a current survey study just to understand some of those experiences. You know, we'll certainly share that link with everyone. For those that have not had a chance to fill that out, that'll be very helpful information. But in addition to that, let me share a bit what we know. So first, there's a lot we don't know, but there's tremendous discussion and coordination between sharing those experiences between Asia, Europe, the US, and other areas that have been affected. We certainly are concerned about COVID and any person at the current time. You know, as we've seen with some of these secondary waves, You know, even 25-year-olds that don't have other medical problems can get very sick from COVID. You know, so this is something we clearly are advising everyone to take the current recommendations: wear your mask, wash your hands, social distancing. You know, avoid all of the politics being drowned out about this. Wash your hands, wear your mask. You know, these things clearly help. As it relates specifically to MPN patients, I think we have the greatest concern that patients with MF may have a higher risk, although we don't know that beyond a shadow of a doubt, but there is that concern. Regarding MPN medicines, we largely have not been dramatically altering the medicines that patients have been on because of fear of COVID. In fact, we have been mindful of the patient with an MPN who is well treated is probably much better off than the patient that has stopped their medication and is, you know, less stable with the disease. The other concern we have is that there are increased risks of blood clots in individuals that have been infected by COVID. So do we need to be more aggressive in anticoagulating MPN patients who are affected? That we do not know, but it is an important area of consideration and concern. But certainly would want to hear from Serge and Allison that similarly have, you know, very large practices where this is being seen in a real-time basis.
Yeah, I cannot agree more with what you said. The cautionary note for everybody is like for us as physicians as well, to be vigilant in preventing infections to the maximum possible, washing hands, making sure that we have a social distancing in place and having a mask on until the time improves for us. But right now, that doesn't appear to be happening anytime soon. So we need to continue to be vigilant about this and continue to be preventative as much as we can. And the notion that you said about actually paying attention to be well treated and not to compromise the delivery of the medication, stopping a therapy because you think it may be endangering your immune system or altering it so that you may be possibly more at risk for getting infection or sicker if you get it, that does not really appear to be the case. And as a physician, through American Society of Hematology, in concordance with the European colleagues, and hence actually a knowledge about not changing the therapy and optimizing the therapy so that patients are in best possible shape and in best possible situation with their underlying disease in case there is an infection, God forbid. So I think that is the message, that to stay on top of your disease, to not alter the management. In fact, be more vigilant in not just preventing infection, but controlling your underlying disease as well.
And I would also really like to echo that. It was beautifully put, Serge, and that is exactly the case here. I live in Maryland, which we have the luxury of a great healthcare system and also a very proactive governor who protected us quite well and kept the rules in place for as long as he could in terms of distancing. I've met with so many patients who, of course, had concerns. Does their disease put them at higher risk? And I echo that. I think, well, we all have to be aware of our risks. MPN patients tend to be older, but also they tend to have problems with coagulation, many, and is this going to increase their risk if they got COVID? So I think all of these recommendations are really very relevant. One thing I would like to say is that I really agree with Serge also about not stopping therapy. So many have asked me, does my interferon or ruxolitinib, does that increase my risk of getting COVID, or will I do worse if I get COVID? And I think absolutely the opposite, that these may even be protective if you get COVID, and they should really be maintained. In terms of my own practice, I only know of one MPN patient who acquired COVID, and an 88-year-old wonderful lady who is doing great. She's been on ruxolitinib for many years. And she got COVID because she lives in a Baltimore row house with her son, who 6-year-old son who got COVID, and she got it from him. She was admitted to hospital and was in for a couple weeks, but survived, and we kept her on her ruxolitinib, and she did very well. So I think just attention to detail and to understanding how COVID affects us all, and being very cognizant of how to protect each other is very important.
Wonderful. Well, I wanted to say it's great to hear that there are experiences of people who have gotten through it, and I'm very relieved to know that we have great doctors like you to help our patients, and the MPN Research Foundation generally is so grateful to all of you on the call. I think we're actually after the hour now, so I think we have to wrap this up, but I did want to, again, thank you all the panelists, thank you all patients and caregivers who have contributed information already to MyMPN. Please continue to do so. And for doctors and researchers, please do reach out to us if you'd like to get involved. Researchers in particular, and pharmaceutical companies, if you think that there's some data that might be helpful to you in your efforts to assist MPN patients, please let us know. And as a general commercial, MPN Research Foundation is available and provides all sorts of connections throughout the community and information for people who have been diagnosed. Please do share information about us, and don't be shy about contacting us if you have questions. And we thank you all so much for your participation today. I think it was a great webinar and really helpful to understand what we're doing here at MyMPN and at the MPNRF. Thank you to all of our panelists and to Dr. Malaturno.
Thank you, everybody.
Thank you.
Bye.
Have a wonderful day.
Bye.